Good afternoon. My name is Megan Kelchen, and I'm a pharmacologist in the Office of Research and Standards. And today I'll be discussing the differences between a no difference standard and Q1, Q2 sameness for topical drug products. This presentation reflects my own views and should not be construed to represent the views of the agency. So I'll begin today by explaining the differences between Q1, Q2 sameness and a no difference standard for generic topical products. And then I would like to discuss some examples of differences between a test and reference topical product that may be acceptable under a no difference standard and would therefore qualify for a characterization-based approach. So to begin our discussion today, it's helpful to start by thinking of the Code of Federal Regulations for topical drug products. And what I'll highlight first is that an ANDA for a drug product intended for topical use may include different inactive ingredients compared to the reference product, provided that these differences are identified and characterized, and that these differences do not affect the safety or efficacy of the proposed drug product. In the case of topical solutions or solution-based foam aerosols, a waiver of in vivo BE studies may be appropriate if the test product contains no inactive ingredient or other change in formulation that may significantly affect systemic or local availability. So a test product that qualifies for a waiver of in vivo studies may be Q1, Q2 the same, but not necessarily, as topical products are not required to be Q1, Q2 the same as the reference product. And so we can use this to build a framework when developing a no difference standard as part of a characterization-based bioequivalence approach for these topical products. So before we move into the no difference standard, let's first discuss why we may want to develop a test product that is Q1, Q2 the same to the reference product. Q1, Q2 sameness of a test and reference topical product can mitigate the risk of known failure modes. And these failure modes may relate to irritation and sensitization, formulation interaction with diseased skin, vehicle contribution to efficacy, and stability and solubility of the active ingredient in the drug product. While Q1, Q2 sameness can mitigate all of these failure modes, implementing Q1, Q2 sameness can be challenging for generic topical products, and this is due to a variety of reasons. For example, there may be changes to the reference product over time such that the current reference product that's available on the market has a different formulation than the original reference product. A reference product may not list a nominal amount for certain inactive ingredients in their formulation table. For example, a reference product may use a quantity sufficient amount of Ph modifier in order to achieve a specific pH target. And this can be difficult for a test product to match the nominal amount of this inactive ingredient because the amount of the inactive ingredient used in the reference product can vary from batch to batch. A reference product may also be discontinued and then therefore unavailable for deformulation, which complicates the development of generic drug products. And so because of these challenges in implementing Q1, Q2 sameness, a more sophisticated assessment of the formulation can ensure that the test product is well matched to the reference product. And this is a key point I want to reiterate, that when we are looking at a formulation assessment, we want to ensure that the test product is well matched. And in order to ensure that these products are actually well matched, we may need a more nuanced assessment of the formulation that goes beyond Q1, Q2 assessment. So this leads us to the no difference standard that is recommended for topical products in order to qualify for a characterization-based bioequivalence approach. Under the no difference standard, a test topical product should contain no difference in inactive ingredients or in other aspects of the formulation relative to the reference product that may significantly affect the local or systemic availability of the active ingredient. So let's discuss the key differences between Q1, Q2 sameness and the no difference standard. Under Q1, Q2 sameness, a test product should have the same components in the same concentration as the reference product. And generally, there are potential tolerances of plus or minus 5% difference in the amount of an inactive ingredient between the test and reference product. The no difference standard is based upon the same principles for assessing Q1, Q2 sameness, but it also takes into consideration differences that may have previously been determined to be acceptable based on available scientific evidence. A test product that qualifies for a characterization-based approach under the no difference standard may be Q1, Q2 the same to the reference product, but not necessarily. What the no difference standard does not mean is that any formulation or proposed test formulation would be acceptable for a characterization-based approach. Again, we want to ensure that the test product is well matched to the reference product. So as part of the no difference assessment, we are evaluating whether certain components and compositions of a test product may be acceptable compared to the reference product. And to do this, this is based upon information available to the agency. For example, we may utilize variations of batch to batch of the reference product or changes that were made to the reference product over time. This also may be based on evidence submitted in an ANDA. For example, evidence to demonstrate no difference between the test and reference product in the local or systemic availability of the active ingredient. As previously mentioned, Q1 Q2 sameness when an RLD product is discontinued can be complicated as the product is not available for deformulation. The designated RS product may not be Q1 Q2 the same as the RLD product. When the RLD product is discontinued, a test product may be assessed under the no difference standard with respect to the RS formulation. So I want to spend the remaining time I have today to discuss some specific examples of that highlight the differences that may be acceptable under a no difference standard. And I first want to start with an assessment of ingredient grade. Q1 Q2 sameness of what may be considered a different grades of an ingredient can be complicated. For example, is carbapol 974P, which is polymerized with ethyl acetate, considered to be Q1 the same as carbapol 934P, which is polymerized with benzene? As a second example, is white petrolatum USP considered to be the same inactive ingredient as petrolatum USP, despite having different USP monographs? In the case of carbapol, a change from carbapol 934P to carbapol 974P may have already been considered an acceptable change for the reference product. In this case, a test product that may be suitable if it contains carbapol 974P instead of carbapol 934P. In the second example, white petrolatum is considered to be a more refined grade of petrolatum, so it may be acceptable for a test product to use white petrolatum when the RLD product uses petrolatum. The acceptability of such difference would be determined during the ANDA assessment. Another example is when an inactive ingredient of a reference product contains subcomponents. So Q1 sameness of an ingredient that is comprised of a mixture of subcomponents can be complicated. For example, the reference product may use a proprietary ingredient that's a pre-blended mixture of specific quantitative amounts of subcomponents. For example, the reference product may use a pre-blended mixture that contains 40% of subcomponent A and 60% of subcomponent B. And so the question in the product label of the reference product may list these two components together, or they may list them as separate inactive ingredients. So the question arises of whether a test product can use the same proprietary blended mixture, or if a test product can use the two subcomponents as separate inactive ingredients in their own test formulation. Under the no difference standard, a test product may be suitable if it contains the same quantitative amounts of each subcomponent, rather than using the proprietary ingredient. So in our example, a test product can either use the same proprietary inactive ingredient, or they can add the two subcomponents separately. Let's stay with the assessment of subcomponents for another example. The reference product may use a proprietary ingredient that's a pre-blended mixture of variable quantitative amounts of subcomponents. For example, a reference product may use a proprietary pre-blended mixture that contains between 40 to 60% of component A and 40 to 60% of component B. And it can be difficult for a test product to match the reference product as the amount of each subcomponent may vary between different batches of the reference product. Under the no difference assessment, or under the no difference standard, A test product may be suitable if it contains quantitative amounts of each subcomponent within ranges that were found to be acceptable for the reference product. So in our example, a test product that proposes to use, for example, a 50-50 mixture of subcomponent A and subcomponent B may be acceptable, as this falls within the range for each subcomponent that was determined to be acceptable for the reference product. What about ingredients that are available in different forms? Again, this can be complicated to match Q1, Q2 sameness. As an example, acetate disodium exists in both a dihydrate form and an anhydrous form, and both of these forms are under the same USP monograph. So a test product may propose to use the dihydrate form of acetate disodium that complies with the compendial monograph, while the reference product uses the anhydrous form, which also complies with the same compendial monograph. Under the no difference assessment, a test product may be suitable if it contains a different form of the ingredient with adjustment to the quantitative amounts of pure ingredient and water. So a test product that can use the dihydrate form of acetate disodium when the reference product uses the anhydrous form of the same inactive ingredient, so long as the amount of water in the test product is appropriately adjusted. A similar question arises for ingredients that are available in different purities. And once again, this can be complicated to match Q1, Q2 sameness. The reference product may use alcohol USP, which is comprised of alcohol equivalent to 73.5% weight by weight alcohol as 95% alcohol volume per volume. Under the no difference assessment, a test product may be suitable if it contains a different purity of the ingredient with adjustments to quantitative amounts of the pure ingredient and water. Another example is an assessment of color or fragrance. This can be complicated to match the Q1 sameness of these inactive ingredients. And therefore, a test product may propose to use a different color or fragrance than that used for the reference product. Under the no difference standard, a test product may be suitable if it contains a different color or fragrance than the reference product, so long as the proposed color or fragrance in the test product does not affect the bioavailability of the active ingredient or affect the safety of the drug product. As a final example, let's look at the assessment of a pH modifier. Q2 sameness of the nominal amount of some inactive ingredients can be complicated. As an example, the single point nominal amount of pH modifier in the composition table of the reference product may not reflect the amount that is actually added during the manufacturing process of the drug product. For example, a range of the amount of pH modifier may actually be used, or the amount of a pH modifier may be QS'd in order to achieve a target pH for the reference product. And so this can be difficult for a test product to match the nominal amount of pH modifier, as the amount of this inactive ingredient can vary between batch to batch of the reference product. Under the no difference standard, a test product may be suitable if it does not contain the same nominal amount of pH modifier, as long as the pH of the test and reference product are matched. In all of these examples, the acceptability of such difference would be determined during ANDA assessment. So in summary, certain differences in components and composition may be acceptable for a generic topical product. In general, generic topical products should contain no difference in inactive ingredients or other aspects of the formulation that may significantly affect the local or systemic bioavailability. And a key takeaway point that I want to make here is that when using the no difference standard, we are actually expanding the eligibility for test products that may be acceptable for a characterization-based bioequivalence approach. But at the same time, we are preserving the scientific principles of Q1, Q2 sameness that are critical to mitigate those failure modes associated with bioequivalence. So that brings us to our first challenge question. Which is not true about the no difference standard for a proposed test product formulation relative to the reference product? A, the no difference standard is based upon the same principles for assessing Q1, Q2 sameness. B, a no difference standard is considers differences in components and composition that may be acceptable based upon information available to the agency, or based on evidence submitted in an ANDA. C, a test product that is Q1, Q2 the same as the reference product is an example of a test product that meets the no difference standard. Or D, any proposed test formulation is acceptable for a characterization-based approach, regardless of the differences relative to the reference product. I'll give you a second to think. If you selected D, you are correct. Under the no difference standard, we are considering differences in components and composition that may be acceptable. But this does not mean that any proposed test formulation is acceptable for a characterization-based approach. Again, we want to ensure that the test product is well matched to the reference product. Challenge question 2 relates to the specific examples that I provided with respect to the no difference standard. Which of the following may not be acceptable under the no difference standard? A test product that proposes to use white petrolatum when the reference product uses petrolatum USP. B, a test product that uses a different fragrance than the reference product. C, a test product that proposes to use 12% more than the nominal amount of pH modifier than the reference product. And this increase in the nominal amount of pH modifier is used in order to match the pH of the reference product. Or D, a test product that proposes to use 12% more of the nominal amount of an emulsifying agent than the reference product. The correct answer is D. It is under the no difference assessment, it is not acceptable to have a test product that proposes to use 12% more than the nominal amount of an emulsifying agent in order to qualify for the characterization-based bioequivalence approach. With that, I would like to acknowledge all of my colleagues at the FDA. Thank you for your attention, and I look forward to addressing your questions during the panel session. Thank you.
Hello, my name is Sam Raney, and it's my pleasure to speak with you today about the use of Q3 characterization tests for topical semisolid drug products. I'll offer the standard disclaimer that this presentation reflects my views and should not be construed to represent FDA's views or policies. So let's begin with the concepts of Q1, Q2, and Q3. Now, Q1 refers to the components in a product, and Q1 characterization of a reference product provides a profile of the qualitative components, the ingredients in that reference product. Q2 refers to the composition of a product, and Q2 characterization of a reference product provides a profile of the quantitative formulation composition of that reference product. So Q1 and Q2 refer to the qualitative and quantitative characterization of a product. Q3 refers to the arrangement of matter in a product, and Q3 characterization of a reference product provides a profile of physicochemical and structural attributes that is quintessentially characteristic of that reference product. Now, when we speak about Q3 characterization, we're actually speaking about a variety of specific tests that can be performed, different dimensions can be characterized for that arrangement of matter. And there are 10 dimensions of this Q3 characterization. The first being characterization of the appearance and texture of a product. This is the look and feel of a product. Also of its phase states. Does it have globules? Is it an emulsion? Does it have suspended particles? And the structural organization of matter. If it's a gel, is there a polymer matrix network within that gel? Is it a microemulsion? What's going on within the system in terms of that structural interrelationship of the components of the formulation? And of course, of the polymorphic form of any suspended drug itself, and the rheological behavior, which tells us a great deal about how the different components of the molecular machinery within these semisolid dosage forms interacts with each other in responses to stress. Now, the water activity and drying rate is incredibly valuable to characterize because as a formulation undergoes metamorphosis after it's dispensed and applied upon the skin, the volatile components begin to evaporate, and these can be volatile components, even including water. And as that happens, the concentration of the drug in that remaining residue changes, and so does the thermodynamic activity of the drug in the formulation, and this has an incredibly powerful influence in many instances upon the drug delivery, that rate and extent to which the drug becomes available in the skin. And so characterizing the water activity or drying rate is very important, because even for products that are Q1 and Q2 identical, differences in manufacturing conditions can alter the drying rate and can alter that bioavailability. Now, pH can have an enormous impact also on a variety of aspects within the formulation, whether it's the viscosity or the ionization state of the drug, and of course that pH is dependent upon how well the product is buffered. And for other components that are oleaginous, for example, long-chain hydrocarbons or short-chain hydrocarbons for that matter, in ointments, can tell us a great deal, especially looking at the heterogeneity of those hydrocarbons, can tell us a great deal about the characteristics and the bioavailability that may result from that product, particularly if some of those shorter chain hydrocarbons may be permeation enhancers and may be enriched in a particular formulation. The specific gravity can also tell us a great deal about whether there may be entrapped air within a formulation, where a given volume of formulation may actually represent a different delivered dose because it contains a different amount of drug. And finally, but certainly not the least important by any means, are metamorphosis-related changes. The idea that Q3 characterization is not static, but it tells us about the response of the system, not only to stress, as in the case of rheological characterizations, but also in terms of metamorphosis over a function of time, as it's dispensed from the product, as it's sheared upon the skin, as it undergoes drying, as the thermodynamic activity changes in a very characteristic way, a manner that is characteristic of the Q3 attributes of the product, including perhaps its water activity. All of these things have an enormous impact and can be critical to controlling that rate and extent that is so essential to understanding whether a product is going to be bioequivalent or not. And therefore, each of these 10 dimensions of Q3 characterization systematically mitigates risks of failure modes for bioequivalence. In other words, ways that something can be different between a test and reference product, and that difference in the structural or physicochemical arrangement of matter ends up causing a difference in the rate and extent to which the drug becomes available at the site of action. Therefore, by comparing, characterizing, and matching all these different dimensions of Q3 characteristics, one can systematically mitigate the risk of these failure modes for BE, and thereby support a demonstration of BE. And among these, we break it down into two large subcategories. One is basic Q3 characterization, which refers to characterizing the appearance, texture, phase states, and structural organization of matter, and comprehensive, which is really characterizing everything. It's useful to do this because when we think about what Q3 characterization is useful for, one is that it can support a demonstration of pharmaceutical equivalence. Basic Q3 characterization for both the test and reference products can demonstrate that a test topical product and its reference product are the same dosage form. In other words, they have the same number, type, and structural arrangement of phases, and thereby, by demonstrating that it's the same dosage form, one can support a demonstration of pharmaceutical equivalence. And this is so important for semi-solid topical dosage forms because the nomenclature of these dosage forms, like lotion or gel or cream or ointment, may tell us very little about what the underlying components or arrangement of matter are. One could have an ointment that's an emulsion, or a gel that's an emulsion, or a cream that doesn't have globules, or a lotion that's actually a solution. And so actually characterizing, doing basic Q3 characterization is one of the best ways to truly compare the dosage forms and ensure that the test and reference product are in fact the same dosage form. Q3 characterization can also be very useful to support a demonstration of bioequivalence. In fact, comprehensive Q3 characterizations for both the test and reference products establishes a detailed profile of measurements for Q3 attributes that may potentially be critical to product performance and does so under relevant conditions. Now, matching the detailed profile of Q3 attributes for the tested product and the detailed profile of Q3 attributes for the reference product substantially mitigates the risk of potential failure modes for BE, for bioequivalence, and thereby really helps to support that demonstration of bioequivalence. How do we know this? Well, we've studied it. Here's a set of acyclovir cream products, all pharmaceutically equivalent to each other, but not necessarily bioequivalent. 3 of them are the reference branded product sold in different regions of the world, and 2 are Austrian hybrid or generic products that literature has suggested may have a different bioavailability than the reference products. And what we can see here on each line are the components that compares how the composition matches up among these products. We can see that the US branded product has a distinct set of components and a composition, while the UK and Austrian reference branded products have very similar components, and likewise for these 2 Austrian products. What we found was that the 3 reference branded products, despite differences in their components and their composition, had relatively similar Q3 attributes, and that correlated with relatively similar cutaneous pharmacokinetics. This was distinct from the Q3 attributes of the 2 Austrian non-reference products that were similar to each other and that had a similar cutaneous pharmacokinetics relative to each other, but distinct relative to the reference set in terms of Q3 attributes and distinct in the cutaneous bioavailability and pharmacokinetics as well. Now, this was in vitro. We then took one member of each of these 2 groups, the US reference product and one of the acyclovir products, and evaluated this in vivo as well in an in vivo cutaneous pharmacokinetic study. This was done with dermal open flow microperfusion, where probes were inserted under the skin of subjects. You can see these probes depicted here. And the US reference product was dosed on 2 of the sites as a positive control for bioequivalence to evaluate the reproducibility of the study and to evaluate whether it would accurately find these products to be bioequivalent, and then compared as a negative control for bioequivalence to that Austrian hybrid or generic product. What we see here is that the reference products were in fact accurately and reproducibly found to be bioequivalent within the limits of 80 to 125, and sensitively discriminated for the reference and test product, the negative control for bioequivalence, as not being bioequivalent to each other. In fact, we've seen this with numerous drug products now, multiple drugs and multiple dosage forms. In this case, looking at lidocaine and prilocaine in a cream compared to a gel, where the creams existed as a reference product and a clinically approved generic product that was shown to be bioequivalent based upon clinical evidence. These had similar Q3 attributes and similar cutaneous pharmacokinetics that was different from the Q3 attributes of the gel, which also had distinct cutaneous pharmacokinetics. Now, this was in vitro, using an in vitro permeation test, where the cream had a higher bioavailability than the gel, and the same products were also dosed in vivo using dermal microperfusion to evaluate very similarly that for lidocaine, the cream had a higher bioavailability than the gel. This is for lidocaine, but a very similar profile was seen for prilocaine in both the in vitro and the in vivo. We have yet another data set with metronidazole using creams and gels where the generic cream and the reference cream were found to be bioequivalent based upon clinical evidence, had similar Q3 attributes, had similar rheology, and similarly for the 3 gels, which the reference product had been found to be bioequivalent to the test products, the approved generics, and had comparable rheological profiles to each other, but distinguished on the cream. Likewise for the drying rates for these sets of products, similar within each set, distinguished from each other. And when we look at the cutaneous pharmacokinetics that arose, the gel products, which were found to be clinically bioequivalent, which had well-matched Q3 attributes, were also found to have well-matched cutaneous pharmacokinetics that was distinguished from those of the gel products that was all very similar to each other. In fact, we've seen this in multiple data sets where clinical evidence has demonstrated the bioequivalence of several topical generics that are not necessarily Q1, Q2, or Q3 the same as the reference product, and an expanding body of evidence has demonstrated that these topical generics exhibit comparable cutaneous pharmacokinetics, not only comparable clinical efficacy. So the question arises, when do Q1, Q2, or Q3 differences impact the bioequivalence of topical products, and what may be acceptable differences between a test and a reference product formulation? Well, this brings us to the question of Q3 sameness versus Q3 similarity. We see in all of these data sets a very consistent theme that all of these products are not necessarily Q1 and Q2 the same, but there's no significant impact on bioavailability. So if the underlying matter is not the same, but the Q3 characteristics are well matched, can we say that they are Q3 the same? In other words, if they have the same arrangement of matter, but the underlying matter is not the same, are they Q3 the same? Well, I think this inspires us to think about a slightly different conceptual framework for the way we think about Q3 characterization, and particularly as it applies to the characterization-based bioequivalence recommendations in our product-specific guidances. So allow me to walk you through a novel way to look at this paradigm. And it begins with the question of, is the arrangement of matter the same? And what we say is this Q3 well matched within the range characterized for different batches of the reference product? If yes, then the next question is that underlying matter the same? Meaning no difference, as Dr. Kelchen just explained, no difference that may significantly impact local or systemic bioavailability. If that is also yes, then these products are generally eligible for characterization-based bioequivalence approaches in current product-specific guidances. These are products where the arrangement of matter is the same and the underlying matter is the same. Now, if the arrangement of matter is not the same, in other words, the answer to this question is no, these products are generally eligible for traditional in vivo bioequivalence approaches in current PSGs. And in fact, these approaches are generally applicable across the board here, regardless of the outcomes. But I think a particularly interesting one for us to discuss is what happens when, as we've seen in many of the illustrations in the preceding slides, that the Q3 characteristics are well matched. The arrangement of matter appears to be within the range characterized for the reference product. This is the arrangement of matter for the test product. But it's not necessarily Q1 and Q2 the same. In fact, it may not meet the no difference standard. In that situation, what are the bioequivalence recommendations? Well, evidence tells us that the risk of bioequivalence may be, or the risk of bioinequivalence may be mitigated by characterizations that are specifically relevant, that specifically speak to whatever failure mode for BE arises from that difference. And once evidence can be provided above and beyond the types of characterizations that are recommended in current PSGs, when specific differences occur, that evidence that speaks to mitigating the risk of those specific differences may be relevant to supporting a demonstration of bioequivalence in those instances. So, if PSGs don't already exist, applicants who intend to submit an abbreviated new drug application for a topical product that relies upon Q3 characterization-based bioequivalence approach, for which relevant recommendations have not been published in a PSG, are encouraged to request a pre-ANDA meeting with the FDA to discuss their proposed approach. And what we'd recommend is that these pre-ANDA meeting requests should include sufficient Q3 characterization, at least to determine the number, type, and arrangement of phase states in the reference product, that basic Q3 characterization at a very minimum, and product characterizations relevant to the nature and complexity and the identification of potential therapeutic failure modes for that test product that may arise depending upon the nature of that test product. So in conclusion, Q3 characterization of a reference product provides a profile of physicochemical and structural attributes that is quintessentially characteristic of that reference product. And these attributes may potentially be critical to product performance. Now, basic Q3 characterization can demonstrate that a test and reference product are the same dosage form and thereby support a demonstration of pharmaceutical equivalence. Comprehensive Q3 characterization establishes a detailed profile of measurements for Q3 attributes that may be critical to product performance, and when these are the same for a test and reference product, it can support a demonstration of bioequivalence. Now, here's the challenge question. Which of the following statements is true? Q3 characterization of a reference product provides a profile of physicochemical and structural attributes that is quintessentially characteristic of that reference product. Basic Q3 characterizations can demonstrate that a test topical product and its reference product are the same dosage form, supporting a demonstration of pharmaceutical equivalence. Comprehensive Q3 characterizations matching the detailed profile of Q3 attributes for the test product to the detailed profile of Q3 attributes for the reference product substantially mitigates the risk of potential failure modes for bioequivalence, supporting a demonstration of bioequivalence, or all of the above. And the answer is all of the above. Thank you so much. I'd like to particularly acknowledge so many individuals within the FDA as well as our research collaborators for all the contributions to our work. Thank you very much.
Good afternoon. I'll talk today about a recent research in FDA related to Q3 characterization of topical drug products containing porous microparticles. Before I start my talk, my disclaimer is that this presentation reflects my views and shouldn't be construed to represent FDA views and policies. Porous microparticles that's used in topical drug products are spherical solid particles, water insoluble, having a network of interconnected pores open to the particle surface to provide flow of the external medium in and out of the internal pore structure. The pore structure provides a large surface area and space for inclusion of various active ingredients and some excipients. These microparticles vary in diameter and size distribution, and the pore dimension within the microparticles vary considerably depending on the polymer used to form the microparticles and the diffusive characteristics of the material to be impregnated. They are available commercially with various particle size distribution, pore dimension, volume, tortuosity, and network of the internal space, as well as surface areas. When the drug product containing microparticles is applied to the skin, three scenarios may be used to explain the availability of API to the skin surface for percutaneous absorption. The first scenario is that the microparticle is fusing with the skin surface, at which API is diffusing directly from the microparticle to the skin without releasing the API to the external media of the drug product. And second scenario is that the API is released to the continuous phase of the drug product from which it is absorbed at the skin surface. In this case, the matrix characteristics is the one that's controlling the percutaneous absorption rather than the diffusion from the microparticles. And the third scenario is that the microparticle may be deposited around the hair shaft, and then it is mixed with the sebum, and the sebum may affect the release of the drug from the microparticle and further percutaneous absorption to occur. Two methods are usually used to load APIs to microparticle before inclusion in the topical drug product. The first method is a one-step process. We call it passive loading or liquid-liquid suspension polymerization method, in which drug and polymer is dissolved in a certain solvent depending on the physicochemical characteristics of the polymer and API. followed by diffusion of the organic solvent outside the droplets, and this leads to formation of microparticle in which the drug is embedded. The second method is a two-step process in which it is an active loading. We call it quasi-emulsion solvent diffusion method, where the microparticle is prepared separately, and then it is suspended in a drug solution, followed by mixing for some time and drying all the solvents that used to dissolve the drug, and this leads to precipitation of the drug substance within the pore structure of the microsponges. So what is the advantage of having microparticles in topical drug product? We know that for the conventional drug product, local action and site-specific action or reduced side effect may be expected, which is more convenient for the patient who have difficulty to take the medication by different routes of administration. So it is easily to be administered and can be terminated. We can terminate the drug action easily by washing the skin surface, but irritation may happen to some patient. However, inclusion of microparticle in topical drug products may offer some advantages, such as sustained release of the API, and it can improve the efficacy and safety profile of the drug product. It can offer a prolonged stability as the drug may be suspended in molecular or amorphous state in the microparticle. And this microparticle is offering high internal space for more drug loading capacity. And some of the undesirable properties such as feel or odor of the drug substance may be considerably reduced. And the liquid drug product also can be transformed into free-flowing powder by inclusion into microparticles. So microparticles is offering a lot of advantages, and various drug products were formulated to have microparticles. Critical characteristics of the microparticle may contribute to the in vitro surrogates of demonstration of bioequivalence of these topical drug product. And this, of course, in addition to physicochemical characteristics of the formulation itself. For example, here we are showing some of the Q3 similarities that may be considered when comparing test to reference drug products containing microparticle. For example, drug loading, as well as rheology and pH of the matrix, as well as drug to polymer ratio in the microparticle and polymer cross-linking, particle size distribution of the microparticle and other dispersed drug, as well as morphological assessment of the microparticles, including porosity and porosity parameters such as pore size, pore volume, dimension, surface area, tortuosity, etc., as well as the spatial distribution of API within the microparticle structure and presence of any electric charges at the surface of the microparticle that contributes to its stability and prevents aggregation within the dosage form, as well as physical stability and globule size distribution, and this should be considered for multiphasic topical drug products such as emulsion, as well as presence of API in different polymorphic forms. Some of these Q3 parameters may be mitigated by running IVRT method, where the API release testing from the microparticle as one of the intermediate product and from the finished product may be done, as well as IVBT may be done, and this especially for multiphasic topical drug products, and this to assess drug distribution among various phases of the drug product. And this is done, however, the IVBT is still questionable about its use as a surrogate for API bioavailability from these drug products. So now we can go to one of the case studies that we have at FDA premises about tretinoin topical gels that has microparticle. And the objectives of this study is to compare the physicochemical properties of tretinoin-loaded microparticles prepared in-house or separated from the drug product, and evaluating the effect of physicochemical characteristics of the microparticles on tretinoin release using in vitro IVRT method, as well as in understanding the interaction between tretinoin and the polymeric matrix of the microparticles, as well as evaluating proportionality of tretinoin release, as well as other physicochemical characteristics across 4 strengths of tretinoin gel. Surface morphology of the microparticle drug product as well as tretinoin powder and processed tretinoin was done. Polarized images of the blank microparticle shows that these microparticles were spherical in shape, polydispersed, and free from aggregates. Images of the drug product show that spherical microparticle present in the gel matrix. These microparticles were well separated by the semisolid excipients of the shell, and no excipients were observed. And at the magnification of 200x by polarized microscope, it was not feasible to visualize the surface characteristics of the microparticles. Hence, scanning electron microscope was used for this purpose. Scanning electron microscope images showed that separated microparticles have rough surface characteristics compared to the blank microparticle, and this surface roughness may be formed during the impregnation process in the non-aqueous solvent for drug loading. No obvious pores were observed at the surface of these microparticles, therefore, more specific tools such as mercury intrusion, porosimetry, and PET nitrogen absorption may be used to evaluate the pore volume and specific surface area of the microparticle. Optical images of the separated microparticle didn't show any sign of tretinoin crystallization among or at the surface of the microparticles, and this may indicate that tretinoin is precipitating in an amorphous or molecular state within the pore structure of the microparticles. So we have also evaluated the microparticle and the drug products for entrapment efficiency and particle size distribution. For entrapment efficiency, this is a method used for extracting tretinoin from the drug product and from the separated microparticles, and these are the entrapment efficiency results. So the results show that only 1% of tretinoin was found in the supernatant of the separated microparticle, and this result shows that tretinoin was mostly present within the microparticle of the gel with minimal to no partitioning in the gel matrix. And for particle size distribution, dynamic light scattering was used for this purpose. And the results obtained showed that the blank and the in-house drug-loaded microparticle have relatively small particle size with the majority of the particle were smaller than 30 micrometer. And the separated microparticles and those in the diluted gel product have larger particle size of 70 micrometer, and this data demonstrated that dynamic light diffraction is an appropriate method for evaluating the particle size distribution as one of the Q3 characteristics of the microparticles as an intermediate product and those of the finished drug product as well. The in vitro drug release was also evaluated from the drug product separated microparticle as well as in-house prepared microparticles. So the release data of tretinoin from the drug product and separated microparticle were almost similar. However, significant higher cumulative amount and rate of tretinoin release was observed from the in-house 1% drug-loaded microparticles. And this result can be attributed to the smaller size of the in-house drug-loaded microparticle used. These release data may indicate that the gel matrix has no to minimal effect of controlling the drug release rate, and the tretinoin release is mainly governed by the critical physicochemical properties of the microparticle. And despite having the same particle size distribution, increasing the drug loading in the microparticle led to a significant increase in rates and amount of tretinoin release. This may indicate that tretinoin is more localized at the periphery rather than at the depths of the pore structure of microparticles. And for this reason, Raman mapping or Raman analysis studies was performed to determine tretinoin distribution within the pore structure of the microparticles. This slide shows the data of in-sim Raman analysis of blank microparticle processed tretinoin and the in-house tretinoin-loaded microparticle and separated microparticle from the gel. Two groups of Raman data were generated. The first group was collected from the outer surface of the intact microparticles, and the second group was collected from the inner core of the microparticle after crucisection. and the Raman spectra collected were compared to the Raman spectra of the Planck microparticle and processed tretinoin for a tracking purpose. And as you can see from these spectra that the in-house tretinoin-loaded microparticle, the drug was more associated at the surface rather than the core of the microparticle. As on the contrary, for the separated microparticles, the drug was more localized at the core of the microparticle rather than the periphery of the microparticle. And this result suggests that the washing step during extraction from the drug product didn't remove tretinoin completely from the separated microparticles. However, the impregnation process of the in-house loaded tretinoin-loaded microparticle was positioning tretinoin more at the surface rather than the cores of the microparticles. To further understand how tretinoin is interacting with the polymeric matrix of the microparticles, interaction studies using FTIR, DCC, and XRD were performed. And in this slide, we are only showing the data of FTIR studies. FTIR spectra of raw and processed tretinoin, it shows a characteristic band for strategic vibration of carbonyl group. And when we see the spectra of microparticles, it shows a characteristic band also of carbonyl groups. And the FTIR of the physical mixture, it shows a corresponding band from both tretinoin and microparticle. On the other hand, disappearance of the carbonyl group of the microparticle and shifting of its hydroxyl group, shifting of its hydroxyl band of tretinoin indicates a molecular interaction of tretinoin with the polymeric matrix of the microparticle, and this interaction was done through hydrogen bonding. This interaction may also indicate that molecular dispersion of tretinoin is evident throughout the specific surface area of the microparticle. Therefore, the specific surface area is considered one of the critical Q3 characteristics of the microparticles and the drug product. We further investigated the dose proportionality for different strengths of the gel drug product. And for this purpose, two groups of the gel were prepared. The first group were prepared using different amounts of the microparticles that has the same drug loading, and 4 gel products were prepared for this group. And the second group has also 4 drug products that were prepared using the same amount of microparticles. However, different drug loading was used for each microparticle. These gel products were evaluated for microscopic evaluation. And the microscopic images of group 1 of the gel product show a decrease in the amount of the microparticles in the gel matrices, with decreasing nominal strengths for the tretinoin gels. However, same amount of microparticles were observed in group 2 of the gel product. And for rheology assessment, for both groups, shear thinning behavior of the rheology data were observed. This behavior was more pronounced in group 1 compared to group 2. In group 1, an increase in the shear thinning rate was observed by increasing the percent of the microparticle in the gel product. And this effect was not observed in group 2, as it contains a similar amount of the microparticle. And similar trend was observed for tretinoin amount in either group 1 and group 2. However, it's worth noting that the association of tretinoin to the microparticle network gave this similar correlation. However, it was just a numerical correlation rather than causation, as no interaction of tretinoin with the gel matrix was observed. Similarly, the loss and storage moduli increased proportionally by increasing amount of the microparticle in group 1. However, no correlation could be driven for both moduli in the group 2. And these results showed that manufacturing parameter and microstructure of the gel products containing microparticle can affect the rheological parameters and should be considered during evaluation of the similarity of test to reference product. The drug release properties of these proportional gel products were also evaluated. In both groups, The rate of tretinoin released from the gel were proportional to the nominal strength of the drug product and with acceptable correlation coefficient of about 0.95 were obtained. These proportional studies may be then used as another in vitro surrogate for demonstration of bioequivalence of proportionally formulated intermediate strengths of the topical drug product containing microparticle, especially when it is prepared by various manufacturing methods. So, for my conclusion, these Q3 characteristics of the microparticle have been identified, and the corresponding specific analytical matrices may be used for evaluating them in both intermediate and finished product. The critical physicochemical characteristics in the topical drug products include particle size, specific surface area, porosity parameter, drug loading, and drug microparticle interaction, as well as API distribution and material attributes of the polymer. So if a test and reference product are Q1 and Q2 similar, then these analytical matrices may be used as in vitro surrogates for demonstration of bioequivalence. And we have also seen that tretinoin release from the microparticle is influenced by the critical physicochemical characteristics of the microparticle, as well as tretinoin was molecularly dispersed in an amorphous state within the pore structure of the microparticles, and the rate of tretinoin was proportional along the four strengths of the tretinoin topical. So we can go now to some of the challenge questions. So the first question is that the following are routes for skin transportation of topical microparticle except: first, transcellular transport of microparticle across the skin; second, paracellular transport of drugs released from the microparticle across the skin; third, accumulation of microparticles in the hair follicle for further release of drugs; and fourth, fusion and lysis of microparticle as stratum corneum. So, of course, the first and last one are not true. My second question: which of the following statements is not true? First, amount of microparticle may affect drug release and neurological properties of topical products. Second, topical microparticle may be used to transform liquid drugs into a free-flowing powder. Third, chemical interaction of the drug with the polymeric matrix of the microparticles can be detected by XRD analysis. And fourth, spatial distribution of the drug within the topical microparticle is one of the Q3 similarity parameters that can affect the drug release properties. So the right answer is chemical interaction of the drug with the polymeric matrix of the microparticle can be detected by XRD. XRD is more used to detect the crystallinity rather than chemical interaction. And this concludes my presentation. And these are some resources that may be used to give more information about topical drug products that has microparticle. And at the end of my presentation, I want to acknowledge all OBQ and OG scientists that contribute to the research of microparticle. Thank you for your attendance and listening to my presentation.
Thank you for the very nice introduction. My name is Eleftheria Tsakalozou, and I am a pharmacologist at the Division of Quantitative Methods and Modeling, Office of Research and Standards, Office of Generic Drugs. Together with my colleague, Dr. Khondoker, I will go over challenges and considerations associated with model-based virtual bioequivalence assessments and how these apply to generic dermatological products. I will go over part 1 of this presentation, where Dr. Alam will complete part 2 of the presentation. My disclaimer. Learning objectives for this talk include: describe virtual bioequivalence assessment and its applications towards supporting product development and approval of generic dermatological products; explain the considerations for employing in silico methodologies and performing virtual bioequivalence assessments for these products; discuss the challenges when performing virtual bioequivalence assessments and what are the future directions; discuss the general factors to be considered in dermophysiologically based pharmacokinetic modeling; discuss 2 case studies of virtual bioequivalence approach that highlight product-specific challenges and model development processes. During this part 1 of the talk, we will go over the Bioequivalence Recommendations for dermatological drug products. We will discuss in silico methodologies and virtual bioequivalence assessment approaches through a case example. We will discuss considerations and challenges in implementing virtual bioequivalence. We will talk about how virtual bioequivalence is important and useful for dermatological drug products, and we will end with take-home messages. Studies that are recommended by the agency towards establishing bioequivalence for generic dermatological products include, but are not limited to, comparative clinical endpoint BE studies. However, these studies are relatively insensitive in detecting formulation differences. There's large variability in the observed response, and dermatological products are of modest clinical efficacy. BE studies with PK endpoints are also recommended. However, for most semisolid dosage forms, typically we do not see detectable systemic exposure, and when we actually see quantifiable amounts of the API in the systemic circulation, these may not reflect local concentrations. Also, recently, more and more drug product characterization studies are recommended in our product-specific guidances. Again, this is not an extensive list of all the studies that are recommended for bioequivalence for dermatological drug products. In silico methodologies for generic dermatological drug products can be implemented towards supporting alternative bioequivalence approaches. These in silico methodologies require the integration of information on drug product characterization studies, skin physiology in individuals and populations, as well as information on skin bioavailability. The latter can be obtained using studies such as microdialysis, as well as in vitro permeation testing. All this information is integrated into a computational framework that allows us to predict amounts of the active pharmaceutical ingredient locally in the skin as well as in the systemic circulation. Now I would like to take the opportunity to walk you through a dermal Pbk model that supported the approval of ANDA 211253. This ANDA involved the generic for diclofenac sodium topical gel 1%. The dermal Pbk model supported an alternative BE approach for a Q1, Q2, Q3 similar test product. The alternative BE approach did not include the PSG recommended in vivo comparative clinical endpoint study, and the dermal Pbk model was leveraged for virtual bioequivalence assessments on predicted systemic and local exposures. As you can see on the left, the model was used to predict exposure for the reference in blue and the test product in the systemic circulation, and that is the top panel, and the presumed site of action, and that is the bottom panel. This slide provides an overview of the proposed workflow for virtual bioequivalence implementation. It begins with a verified, validated model. So in the case of the dermal Pbk model with diclofenac, you can see how the predictions shown with a continuous blue line for the reference on the left and the test on the right agree well with the observed data, which is the red solid circles. The next step is to use this verified, validated model for simulating an in vivo study. So PK profiles are generated for the plasma on the left and the synovial fluid, the presumed site of action, on the right. The reference, which is in blue, is compared to the test in red. These simulated PK profiles are then used to perform a virtual bioequivalence assessment, where PK matrices are estimated, Cmax, maximum concentration, and area under the concentration curve for the plasma and the synovial fluid are compared using a statistical test between the reference and the test product. Let's go over some key considerations in implementing a virtual bioequivalence assessment. A virtual bioequivalence assessment is essentially a comparison of simulated PK profiles between the reference and the test drug product. This can take place in the different application sites at different skin layers, as well as the site of pharmacological action, which could be the target site. In the case of the diclofenac dermal Pbk model, these will be the dermis and the synovial fluid. The comparison can also take place in the systemic circulation. In our case, that would be the plasma. PK matrices that are used for BE statistical analysis include Cmax or Amax and AUC. The overall shape of the PK curve, which includes the time to maximum concentration, the absorption and the elimination phase, are also considered during model validation. Virtual bioequivalence for dermatological products can be used to assess exposure locally and systemically for the reference and the test product. These can be extremely important when biological samples cannot be collected and when healthy or diseased populations cannot be studied. Virtual bioequivalence approaches can also be used to bridge drug product quality and bioequivalence, define a safe space for clinically relevant quality attributes, and thus justify deviations on quality between reference and test products. And finally, these approaches can be used to inform decisions throughout the entire life cycle of a drug product. Now let's discuss the challenges in performing a virtual bioequivalence assessment. These challenges may pertain to the study design, parallel, crossover, fully or partially replicated, as well as the study population in terms of the age, the gender, the race of the virtual subjects, in terms of correctly capturing special populations as well as diseased ones in the model, and deciding on the sample size for a specific population. Accounting for inter- and intra-subject variability in developed models can be a challenge in performing a virtual bioequivalence assessment. Current mechanistic modeling approaches provide a framework for rationally assigned variability to relevant model parameters. These can be skin physiology parameters, the different application sites where the product is applied, the virtual population, as well as the drug product characteristics and their impact on local bioavailability. Approaches towards accounting for variability may include differentiating it from uncertainty, which is of unknown origin and may be associated with biological methodology. Sufficiently verified and validated models for their intended purpose should be used for virtual bioequivalence assessments. It is important to identify and adequately characterize sources of variability, understanding the different biological processes, experimentally studying relevant sources of variability, and accepting that it may not always be feasible to characterize those sources of variability. In these cases, sensitivity analysis may be an important tool that can be used to identify impactful parameters and assign variability on those under the key assumption of no model misspecification. In summary, modeling and simulation approaches with a virtual bioequivalence assessment component can be used to support product development and approval for dermatological drug products. The thermal Pbk model, developed for diclofenac topical gel, highlights how a model can predict the impact of drug product attributes on local bioavailability. Challenges in the implementation of virtual bioequivalence assessments were recognized and discussed here. Research and collaboration among academia, industry, and regulatory agencies is necessary to tackle those issues. Modeling and simulation approaches coupled with a virtual bioequivalence assessment can be used to support an ANDA. Early interaction between industry and the FDA should be initiated through the pre-NDA meeting request program under GDUFA 2. This completes part 1 of this talk. Please join me in welcoming Dr. Alam with part 2.
Thank you for the nice introduction. My name is Khondoker. I am a pharmacologist working in Division of Quantitative Methods and Modeling within Office of Research and Standards, Office of Generic Drugs. Disclaimer of my talk. Today I will be discussing general expectations on dermal Pbk model and our modeling platform. In the second part of my talk, I will be discussing product-specific challenges and consideration in virtual bioequivalence approach. My talk will be supported with 2 case studies. In this slide, I will be discussing key elements we should consider during the development of dermal Pbk model. An ideal mechanistic dermal Pbk model should reasonably describe the skin physiology. The model should capture key formulation attributes. It is particularly important if the model is going to be used for virtual bioequivalence. For model parameterization process, suitable informative datasets such as IVPT studies or skin biopsies or other form of studies may be used to inform model parameters or define model parameters. We may need to make some assumption on model input parameters, particularly when there is no reliable method is available to obtain the parameter value experimentally. However, our assumption should be reasonable, and whenever possible, it should be justified with supporting evidence. The model predictive performance should be assessed with a wide range of topical drug products. Once you incorporate all these important components, we might have a mechanistic dermal Pbk model in which we have higher confidence. Applicants are encouraged to follow best practices when developing dermal Pbk model. The guidance for industry on physiologically based pharmacokinetic analysis format and content is an excellent source. I have listed 2 papers from FDA authors to learn more about Pbk model development and virtual bioequivalence approach. Case study 1. Here I will be discussing product-specific challenges of clindamycin phosphate topical lotion. This is an oil in water emulsion indicated in the treatment of acne vulgaris. Our current PhD recommends comparative clinical endpoint bioequivalence study. However, this study may not be sensitive enough to detect formulation differences. We may see large variability in the observed response. The study is also costly as it requires a higher number of subjects, and the entire drug development process may be also lengthy. These challenges can be overcome with Pbk model-based virtual bioequivalence approach. Agency welcomes innovative approach, and the advantage of Pbk model-based approach is that we can estimate the bioavailability in local tissue. This approach is also cost-effective and may offer faster drug development process. Now let's talk about how we can develop mechanistic dermal Pbk model for clindamycin phosphate topical lotion. This is an unique product following topical application of the lotion. Most of the clindamycin phosphate is hydrolyzed to clindamycin base by enzymatic reaction on the skin surface and within the skin. So the model structure should capture this hydrolysis process. Partition coefficients and diffusion coefficients are 2 important model parameters that determines the permeation of API through the skin. Our model structure should capture the dynamic permeation process. The value of partition and diffusion coefficients may be informed by IVPT study or other suitable form of experiments such as skin biopsy, microdialysis. Now let's talk about metamorphosis of the formulation. Vehicle of clindamycin phosphate evaporates rapidly from the skin surface. This may change the extent of phase distribution of API, as you can see from the figure. This can also increase the permeability of the API by increasing the thermodynamic activity of the drug molecule. Our model structure should capture this dynamic process, and to assess the effect of vehicle evaporation, we may conduct IVPT study under occluded and non-occluded conditions, and we can incorporate this data into our model, and the model's predictive performance also can be verified with this experimental data. Clindamycin phosphate topical lotion is a complex system. It is an oil in water emulsion. The size of emulsion globules and the distribution of API in 2 phases may potentially impact the permeation of drug molecule into the skin. Our model structure should capture these 2 key parameters, and these parameters' value should be informed with experimental value. Other important model parameters related to formulation attributes such as formulation pH, apparent viscosity, specific gravity should also be informed with experimental value. Now we have mechanistic dermal Pbk model for clindamycin phosphate topical lotion. The next step is to verify or validate the model or modeling platform. My colleague Dr. Tsakalozou has already discussed this aspect very nicely in her presentation. Also, please remember that for virtual bioequivalence, we also need to incorporate population or inter-subject variability and intra-subject variability into our simulation. Once everything is considered into our model is ready to predict the bioavailability of test and reference formulation in different layers of skin as well as systemic compartment. Case study 2. In this case study, we will consider a hypothetical topical cream in which the API has low skin permeability. The pharmaceutical form of this hypothetical cream is oil in water emulsion, and presumed site of action is skin. Also assume that the agency allows in vitro characterization-based bioequivalence approach, which includes IVPT study. However, due to low permeability of API, conducting the IVPT study may be challenging. As a consequence, the overall cost of the IVPT study may be increased. Also, the entire drug development process may be delayed. Pbk model-based virtual bioequivalence study may be a potential solution. Agency consider this as an alternative approach. Under this alternative approach, we still need to characterize, test and reference drug product with suitable in vitro studies. In lieu of conducting pivotal IVPT study, the Pbk model may be used to perform a virtual bioequivalence locally and systemically between test and reference drug products. Now let's talk about the Pbk model development process. The general expectations on dermal Pbk model and or modeling platform still remain the same as discussed under case study 1. The model should capture key formulation attributes as listed here. Important model parameters such as partition and diffusion coefficients to describe the permeation of API through the skin may be informed by IVPT study or skin distribution study such as skin biopsy or microdialysis study. Question is, can IVPT study still be used to inform model parameters? Yes, the study doesn't need to be pivotal in nature. However, we expect that the method is validated. Once the model is validated with IVPT study data or other suitable form of experimental data, and the model is informed with key formulation attributes, the Pbk model is now ready to simulate virtual IVPT profiles for test and reference drug products for a given number of donors. To summarize my talk, Pbk modeling and simulation approaches may be used to provide insight on the effect of important formulation attributes that may influence the skin permeability of API, to support alternative bioequivalence approach developed to address challenges with IVPT studies. My acknowledgements. This is a list of Gadufa-funded research projects supporting the dermal Pbk modeling and simulation work. If you are interested, please check out the link to learn more about our research activities. I would be happy to take any questions during our panel discussion session. If you have any follow-up question, you can also reach out to us at the email IDs listed here. Challenge question 1. Virtual bioequivalence for generic dermatological drug products can be used to support bioequivalence assessment between reference and test drug products, bridge bioequivalence and drug product quality, inform decisions throughout the entire life cycle of a drug product. All of the above? Answer is all of the above. Challenge question 2. What factors should be considered towards developing a dermal Pbk model to be used in a virtual bioequivalence approach? Model or modeling platform reasonably describes the skin physiology. Informative datasets such as clinical and or IVPT studies are levers towards informing key model parameters. The formulation attributes are accurately captured. The predictive performance of the Pbk modeling platform is adequately assessed. All of the above? The answer is all of the above.
Thank you to all of our presenters for the great presentations. We'll now enter into our Q&A panel comprising of our session speakers and introducing Dr. Markham Luke, who serves as the FDA Supervisory Physician and Director of the Division of Therapeutic Performance 1 in the Office of Research and Standards and OGD. DTP1 is responsible for facilitating pre-application development of complex generic drugs by conducting and promoting regulatory science research to establish standards to ensure therapeutic equivalence of new complex generic drug products. Now we'll turn to the questions that came in. The first group of questions will be directed to Dr. Megan Kelchen. And here is the first question for Dr. Kelchen. Does the exact quantitative composition, or Q2, need to be determined for no difference?
Thank you for the question. As mentioned in the presentation, the no difference standard is really rooted on the same principles as Q1, Q2 sameness. And so a test product that is Q1, Q2 the same may be considered to be acceptable under the no difference standard. However, for example, a test product that uses, for example, more than the nominal amount of a pH modifier in the proposed formulation in the RLD product may not be acceptable under a Q1, Q2 standard because it doesn't meet the Q2 sameness, but it may be acceptable under the no difference standard if the test and reference pH are matched. So under the no difference standard, a test product may not exactly be Q2 the same as the RLD product, but it may still be eligible for a characterization-based approach. Thank you.
Thank you for responding to that question. We have another question for Dr. Kelchen, and here's the question. The no difference standard would apply to what group of topical products? Topical dermatologicals only, like gel, ointments, creams, lotions, or dental oral gels as well?
Thank you for the question. This is a great question. I want to first note that the no difference standard is not applicable for products that are required to be required by the regulations to be Q1, Q2, such as the products that were discussed at yesterday's session. The no difference standard discussed in my presentation was primarily focused on locally acting semisolid topical products for for which Q1, Q2 is not required by the regulations and for which bioequivalence is demonstrated using a characterization-based approach. And this may include topical dermatological products such as gels, creams, and lotions, or semisolid vaginal products such as vaginal gels. So a product that does not meet the no difference standard may utilize a different approach, such as a comparative clinical endpoint study to demonstrate bioequivalence. In terms of other drug products, at this time, our PSGs do not recommend a no difference standard for some of these other products. However, if you have additional questions related to whether a no difference standard is applicable for a specific test product, I recommend submitting a controlled correspondence or pre-ANDA meeting request. Thank you.
Thank you for responding to that question. We have a few more questions for Dr. Kelchen, and here's the next question. How can I get feedback from the agency on whether my proposed test formulation meets the no difference criteria?
So this is a great follow-up question to my last answer. If the PSG for a specific drug product recommends a characterization-based approach, then an applicant can submit a controlled correspondence to determine if their proposed formulation for the prospective generic drug product is suitable for the characterization-based approach that's described in the PSG. If a PSG is not recommended, does not recommend a characterization-based approach, a pre-ANDA product development meeting request can be submitted. And when submitting that pre-ANDA meeting request, we advise that you include both the proposed BE approach and the proposed formulation. And at that time, we can provide comments regarding the acceptability of your proposed formulation for your proposed BE approach. Thank you.
Thank you for responding to that question. We do have another question for Dr. Kelchen. Does the no difference standard apply to both locally acting products and systemically acting products?
So the focus of my talk was the locally acting drug products. At this time, our PSGs for products that are systemically acting in nature, for example, transdermal gels, do not recommend a no difference standard. However, again, if you have a specific question about a specific drug product, I recommend following up with communication from the agency. Thank you.
Thank you for responding to that question. Another question for Dr. Kelchen. How does the no difference standard expand the eligibility for a characterization-based approach?
So the no difference standard takes into considerations of Q1, Q2 sameness, but it also takes into differences that may have been previously been determined to be acceptable based on scientific differences. For example, the no difference standard may consider variations between batch to batch of the reference product. So because the no difference standard takes these differences into consideration, the no difference standard is a little bit more of a nuanced assessment of Q1, Q2 sameness, and therefore a test product that may not be exactly Q1, Q2 the same, but still falls in this no difference category, may be acceptable for the characterization-based approach. Thank you.
Thank you for responding to that question. We have one final question for Dr. Kelchen in this round. And here's the question. How does the agency determine what the no difference criteria is for a particular product?
So this is really product specific. We look at the RLD product and, for example, any changes that may have been made to the RLD product, and we also consider what differences have been made or what changes have been made to the drug product over the lifetime of the product. So as I've mentioned in my previous responses, we're really focused on a Q1, Q2 assessment, but we do take into those product-specific considerations. Thank you.
Thank you for responding to that group of questions. Now we're going to move on to Dr. Sam Raney, and here's the first question. Can no difference be evaluated by in vitro tests like IVRT, IVPT, and rheological studies between test and reference product? What is the correlation between Q1, Q2, and Q3?
Well, thank you. It's a good question. So think about no difference, specifically as meaning no difference that's going to cause a local or systemic difference in bioavailability. So in order to demonstrate that one satisfies the no difference criterion, an IVRT or Q3 characterization, these provide supportive evidence, and IVPT can provide direct information about whether potential differences between a test and reference product have affected the local bioavailability, but an IVPT study would not provide information about whether there could still be an impact to the systemic bioavailability. And so the no difference standard should really be thought about as very similar in its fundamental concept to a Q1, Q2 type standard. But there are instances where topical products are a little bit unique, and so applying that Q1, Q2 standard doesn't always fit. There's many products that have been regulated under a Q1, Q2 standard for many years, and all of the generics that have been approved have the exact same formulation, or Q1 and Q2 the same, compared to the original RLD. And even if that original RLD is then discontinued and a new reference standard is established, a Q1, Q2 assessment is now made relative to that new reference standard, and it is the exact same formula, components, and composition. Now, when we talk about no difference, one of the things that we have to make an allowance for with topical products is that when the RLD is discontinued and the new RS is in place, that RS may have evidence, potentially clinical evidence, bridging the bioequivalence of those products, but their formulations may not actually be the same. And so there are conventions where we've utilized the no difference standard, where you would say, well, Q1, Q2 sameness would require you to demonstrate that your test product matches the components and composition of the RLD, which is now discontinued, which you can't even deformulate anymore, and which is different than the current RS. So in this way, that would sort of be a blockade for being able to use a characterization-based approach. The no difference standard takes into account some of these things, but specifically, one doesn't just use IVRT or IVPT. One tries to then understand where's the evidence coming from to demonstrate that there is no difference in systemic or local. And so there are instances where, for example, the PSG for a product may recommend characterization of components, composition, Q3 attributes, IVRT, IVPT, and a systemic PK study. Now, in those instances where there may be small differences, where a particular ingredient may, let's say, exceed plus or minus 5%, maybe at plus or minus 6%, but you have an IVPT study that shows bioequivalence, and you have an in vivo PK study that shows bioequivalence, that's the kind of evidence where you would say, I now have evidence that there is no impact at being plus or minus 6%, no impact on either local, you have IVPT data for that, and no impact on systemic, you've got in vivo PK data for that.
Thank you for responding to that question. We have another question for Dr. Raney, and here it is. How do you address Q2 when you are testing for an excipient which exhibits significant lot-to-lot variability in the RLD greater than 5%? Do you set the target at the nominal range?
So if I understand this question correctly, the question is, rather than having a point value for the quantitative amount of a given ingredient in a test product, I think the question is asking whether there can be a range. That isn't what I'd recommend. There's a lot of expertise that goes into how one determines what the nominal composition of a test product should be, and it may happen that in deformulation that you may find differences in your measured value for a given ingredient. Now, is that because of imprecision in the test method that that's difficult to know what the particular case that you're dealing with is? But one of the things you want to understand is, what are you seeing in the reference product, and most importantly, what is it that when you use different specific amounts, you may have to test out using different specific amounts of an ingredient in that range in your test product, manufacturing it using your process, and seeing what the Q3 characteristics are, and then making a decision of what your test product is going to be. And that's really what you would typically, in this case, perhaps come into a pre-ANDA meeting saying, we believe that we would match this by having this specific, not a range, a specific nominal composition for your test product, and you might have 3 different options, and request some clarification from the agency about whether you would qualify for a characterization-based approach, or if you're really having trouble, you could potentially have a pre-ANDA meeting with the agency to discuss your data, your deformulation data, and your challenges.
Thank you for responding to that question. We have another question for Dr. Raney. Could you please clarify whether, in your opinion, that unless the generic topical can demonstrate its Q3 sameness similarity to the RLD, it may be difficult for FDA to accept alternative approaches, for example, in vivo PK study, to establish BE other than a successful clinical endpoint trial?
So I believe this question is referring to the slide that I had with the arrows. Where the top, the first question was, is the arrangement of matter the same? In other words, is the Q3 well matched? And there's a scenario there where the Q3 is well matched, but there's some difference in the test product relative to the reference. Some difference that means it's not going to qualify for the no difference standard. So maybe it's at plus or minus 6%, maybe it's some other reason. In those instances, the concept is that there's a pathway forward if at least the arrangement of matter is the same, the Q3 is the same, and then you might utilize some evidence specific to that difference. Perhaps it's in part referring to the inactive ingredient database, perhaps in part some modeling and simulation. One might utilize an in vivo dermal OFM or dermal microdialysis study. So there are a lot of options potentially in situations where at least the Q3 characterization finds them to be matched. In situations, I think the question is asking, in situations where the Q3 is not matched, what are the recommendations there? And I wouldn't say that the only option would necessarily be a clinical endpoint study. However, I would kind of look at it the other way around to say that, there is a substantially greater opportunity for being able to utilize alternative approaches if you can at least match the Q3 characterization, because we know that one of the ways that the components and composition exert their effects on bioavailability is through the molecular machinery of these dosage forms. The Q3 characterization helps us to mitigate the risk thereby of those failure modes. And so I think that's the encouraged road forward, but it doesn't mean that if there is a small difference in rheology that's outside the range characterized for the reference product, that the only option would be a clinical endpoint study. No, I would say not necessarily.
Thank you for responding to that question. We have another question for Dr. Raney, and here it is. Can you please direct us to resources to use to develop a generic drug development program?
Well, I think the most direct resources would be FDA's product-specific guidances. We have on FDA's website a, a searchable site that contains that if you can search, if you're looking for a particular product, a drug in dosage form, that we have product-specific guidances that have recommendations for how to approach the drug development for that, or at least the bioequivalence approach for that. As far as the drug development process itself, I'm not sure that there are specific references to which I can point there.
Thank you for responding to that question. We've got a few more questions for Dr. Raney, and here's the next question. What is the value of basic Q3 characterization alone, since all the components of basic Q3 characterization are included in comprehensive Q3 characterization?
So it's valuable because demonstrating bioequivalence for certain products may not involve a comprehensive Q3 characterization. This may happen for products where there's a clinical endpoint study or AT-rated products. So in these instances, relying simply upon the dosage form nomenclature may not be very helpful, because topical dosage form nomenclature can have some ambiguity. So, for example, a product that's called a lotion may actually be a solution, or a product that's called a gel may actually be an emulsion. Product that's called a cream may not even have globules. So the basic Q3 characterization provides the specific evidence to demonstrate that the test product is truly the same dosage form as the reference product, and there are situations where just demonstrating pharmaceutical equivalence independently of demonstrating bioequivalence using Q3 becomes quite valuable.
Thank you for responding to that question. We have one last question for Dr. Raney in this round, and here it is. How can we characterize oleaginous components for a simple petrolatum ointment?
So the USB monograph for petrolatum contains a series of tests that can be performed on petrolatums, and these actually help to establish whether a specific source of petrolatum is within grade. So we've conducted research where we were able to perform these tests with finished dosage forms that were ointments that were predominantly petrolatum. They might have a little bit of mineral oil or something in them. And we were able to compare the quantitative results from these tests for the test and the reference product. So that's one way to characterize the oleaginous components. But I think a particularly valuable way in our research has been to characterize to do actually a comparative assessment of the hydrocarbons that are in the test and the reference product in these oleaginous components. So we're looking here at the distribution of different types of hydrocarbon species in the test and the reference product, and looking at that distribution, that profile of the hydrocarbons, is perhaps one of the most valuable ways to be able to characterize the oleaginous components in a tested reference product.
Thank you for responding to that question. The next round of questions will be directed to Dr. Ahmed Zidan. And here's the first question for Dr. Zidan. What are some of the materials commonly used to form the microparticles used for drug loading for topical products? Do certain materials offer certain advantages over others, or is the choice of material primarily driven by characteristics of the API with which the microparticle will be formulated?
So thank you for the nice question. And to answer this question, it is not a matter of the material itself. It is a matter of the quality of the microparticles that are expected to have, or it is a matter of the quality attributes of this microparticle, as well as it is a matter of the formulation process that we are going to use to impregnate it with API, or even to formulate the microparticle itself. Some of these materials includes non-erodible mesacrylate cross polymers, and these polymers vary so hugely in physicochemical characteristics that can affect the physicochemical properties of the microparticles that is formed. For example, the pore structure, as well as tendency to interact with certain APIs, so that the interaction may be so strong that not allow API to be released, not to be released from the microparticle structure. So it is a matter of the quality of the microparticle, and this comes after the safety of the microparticle material. So the non-biodegradable and inert microparticle is a key for choice of the appropriate microparticle material to be used, as well as formulation to be incorporated in.
Thank you for responding to that question. The next question, we'll direct it over. We're switching panelists to Dr. Eleftheria Tsakalozou, and it's referencing on slide 8, the predictions of the model do not agree with the observed data. How can this pass validation criteria? If they do, what are the validation criteria that would accept such large differences?
Thank you very much. This is a great question. Due to time limitations within this presentation, I was not able to share the entire validation process that was performed by the applicant and also by the agency supporting the Pbk model for ANDA 211253. Just to inform the audience, this and the individual that asked the question, this extensive validation process involved assessment of the performance of the platform that was used to develop the model, and it also involved the development of dermal Pbk models for products that included diclofenac sodium, but were not the emulsion, the reference product. So when it comes to the modeling, the performance assessment of the modeling platform, the applicant provided dermal Pbk models for more than 10 drug products. Overall, the performance of the platform was deemed acceptable. And the applicant also offered dermal Pbk models, developed dermal Pbk models for diclofenac sodium solutions, gels, and emulsions. So the agency considered all this huge amount of information together to assess whether the performance of the platform and the performance of the model that was specifically developed for the reference product would be acceptable. And our overall assessment was that the model was acceptable and suitably validated and verified for regulatory decision making. Another important consideration when going over model validation in general is to consider trial-to-trial variability that we often see when we are looking at PK data from different sources. And this is also considered on top of inter-subject variability, which was considered during the performance assessment for this fit-for-purpose model. So under all these considerations, we concluded that the performance of the models for the reference and the test products are acceptable, that these models are suitably validated for regulatory decision-making. And my slides also contain reference to a paper where we went over the entire validation and verification process. So I do encourage the audience to go over this paper because I think that a lot of questions will be answered. Thank you.
Thank you for responding to that question. We have another question for Dr. Tsakalozou, and here's the question referencing again on slide 8. There is no apparent TR difference in synovial fluid versus plasma exposure. I presume that this is due to the fact that in the Pbk model, it assumes that synovial fluid and systemic concentrations are related to one another, an assumption you do not want to make in the introduction. Would the Pbk model be able to predict different synovial fluid exposure when systemic concentrations are the same? If not, then what is the added benefit of the model versus a systemic PK only study?
Thank you for this question. So apparently slide 8 was very, very popular. This is another good question. And I would like to thank the individual who asked this question for this opportunity to clarify a couple of things on dermal Pbk modeling. So when we are developing a physiologically based pharmacokinetic model, on skin layers and the systemic circulation are connected and they're communicating with each other. And this is the same as it is happening actually in vivo in our bodies. The main goal of the model is to describe the in vivo situation. So the assumption, as it is pointed out in the question, is correct. We do have a communication and interplay between the concentration, the amounts of the API in the systemic circulation and the amounts in the skin layers. In my introduction, I said that these two amounts of the API and amounts of the systemic circulation may not always reflect amounts in the systemic circulation. So there is a subtle difference between those two. Coming back to these two statements. So coming back to the Pbk models, I want to make sure, I didn't have the time to go over that during my presentation, but I want to make sure that the audience is aware that these Pbk models are very sophisticated. They account for, even for the potential for redistribution of the API from the systemic circulation into the dermis. They describe skin permeation of the API through the skin as it is impacted by blood flow in the dermis and in the muscle, for instance. But blood flow vascularization is not present in the stratum corneum or the veil epidermis. Permeation is also impacted by protein binding. The physicochemical properties of the API in the different skin layers is also taken into account. So all these physiological processes that are present in the skin determine API permeation through the different skin layers. And these physiological processes, along with the differences in drug product quality attributes, such as apparent viscosity, pH of the formulation, globule size and particle size distribution of the API within the formulation, are impacting API permeation. So these models account for the interplay between a drug product quality attributes, and the physiology. And on top of that, model predictions reflect intersubject variability, which may depend on gender, race, the location where the product is applied, so chest, abdomen, etc. And in all that, we take into account variability between trials. So it is possible that predictions, so there is definitely an interplay between between the skin layers, amounts of the API in the skin layers and in the systemic circulation. But that does not mean that these predictions in these two distinctly different locations in our body will be the same. The model does not make this assumption. It does not operate under this assumption because this is not a valid assumption in vivo. So to conclude, when we're using a Pbk model, model predictions on local bioavailability reflect drug product specific quality attributes. As these predictions may or may not be the same between the reference and the test product. And what a model-based virtual bioequivalence approach can provide is actually statistically assess how different the reference and the test product are, and evidently answer the question if they're bioequivalent or not. And this is extremely important when, for instance, we cannot recruit a high enough number of subjects that will allow a bioequivalence assessment by sampling the synovial fluid, for instance. So there are limitations in the recruitment of subjects that are needed for a statistically robust conclusion. And in certain cases, we don't have systemic PK available because the API is not detectable in the systemic circulation. So the model there plays an important role. And finally, it is possible that we cannot access the target, the site of action, due to ethical and feasibility limitations. So in all of these cases, a dermal Pbk model, along with in-depth characterization of the reference and the test product and any other studies that are deemed necessary for risk mitigation, can be used to eliminate a significant knowledge gap, which is what are the local API amounts at the site of action. Thank you.
Thank you for responding to that question. We're now going to move to our next panelist, Dr. Khondoker Alam, and here's a question for Dr. Alam. How is the intra-subject variability estimated for the Pbk model?
Thank you for this question. So this is very nice question and one of the complex question to answer. Let me talk about the inter-subject variability or population variability first. So we have a relatively rich data set to understand that what are the physiological parameters or factors are involved in the disposition of a topical drug product. For example, what are the variability we have seen in the pH of the skin surface, and what are the variability we have seen in the thickness of the different skin layers. How many skin layers are there in different populations. So there are rich data sets we can look into published literature, and we can incorporate those variability into our model. Now, when we are considering the intra-subject variability, so it becomes a little bit complex because it depends on how your formulation or your drug product is dispositioned by different physiological factors. What are the physiological factors particularly involved in the disposition of your drug product? For example, some drug product may be very sensitive to the skin pH. So are you anticipate any change in skin pH in two different occasions? Or are you anticipating that skin hydration may be influencing the disposition of your drug product in two different occasions. So these are few things we need to consider, and also the entire overall approach or incorporating inter or intra-subject variability depends on how you are proposing your virtual bioequivalence approach. You may also conduct a small pilot scale in vivo study to incorporate or to understand the nature of inter and intra-subject variability. So that is another option. So depending on the approach, you have to decide that what kind of how you will proceed with incorporating these two parameters in your model.
Thank you for responding to that question. We'll give Dr. Alam just a moment to adjust his mic, as we just received one more question. It'll be our final question for Dr. Alam. And here's the question. What type of data is necessary for the development of the Cleocentine lotion model? What type of data is necessary for its validation?
Yeah, this is another good question. So I have described this into my presentation briefly. So ideally, the dermal Pbk model of any topical drug product, including clindamycin phosphate lotion, should be informed with formulation-specific parameters such as pH, viscosity, API solubility, distribution of API, if it is emulsion, size distribution of emulsion droplets, and if there is any metamorphosis event or vehicle evaporation event, it should be also captured into our model structure. Now, coming to the second part of your question, what type of data is necessary for the validation of the model? So we can leverage different type of data for validating our model. For example, skin distribution data such as skin biopsy, microdialysis, or quantification of API in skin layers using IVPT setup can also be leveraged for the validation of the model. So also it is also important to understand that if any of this data is not utilized for the model validation process, so this data can also be used for defining certain model parameters such as partition or diffusion coefficient of API. And also systemic PK data sometimes can also be used for validating the predictive performance of the Pbk model. So it is important that experimental condition also adequately captured in our model while validating the predictive performance of the model.
Well, that's all the time we have for questions in this Q&A panel. We certainly would like to thank all of our presenters and panelists for answering numerous questions that came in once again. We'll now enter into our short break time and return at 3:20 p.m. Eastern Standard Time. Enjoy your break.